Welcome to online participants of the aspirants.
Okay, welcome, my dear friends. So today's most important topic we're going to deliver by eminent resource person, Dr. G.BHAGEERATH sir, Assistant Professor, Head Department of Botany, GDC, Rangasahipet, Warangal District. So, my dear friends, it is most important topic for all competitive exams, especially uh, TG set, AP set, CSIR, NTA, CSIR, UGC, NET also. So, and also other competitive exams, CPGET 2025, other competitive exams. So, listen carefully, my dear friends. Uh, in this regard, we would like to invite Dr.
Hello.
Please.
Okay, your voice is break, but even though I will start. So yesterday in our online class, we discussed about various type of gene actions taking place in a chromosome. So today our topic is linkage and crossing over. So what is meant by linkage? Actually, how it is related with the previous chapter. In the previous chapter, we can observe when a cross made between the two parental plants which exhibit the respective characters. Along with parental characters, we can observe different type of combinations in different ratios. But later we can understood that in any crossing, we can analyze the progeny. we can analyze the progeny. We can analyze the progeny. Most of the characteristics of the offsprings are related to the parents. Somewhat few characters are dissimilar with the parents. Why such type of tendency is observed in many plants? That is nothing but it indicates linkage phenomena in genes which are arranged on the same chromosome. Are you getting my point? When we cross any two parental plants with respective features, in the progeny, many of the progeny members are shown parental characters only. That is nothing but a kind of a link phenomenon observed in between the genes that is indicates linkage. So today we'll discuss about linkage topic. What is meant by linkage? Linkage is nothing but tendency of genes to remain together in their original combination during inheritance is called as linkage. So the genes are carried through the gametes. At the time of sexual reproduction, meiosis takes taking place and two homologous chromosomes are separated. These two homologous chromosomes containing genes which are arranged on a sequential order. So whatever the genes are very closely arranged and together, they inherited togetherly and they carried into gamete. So when the gametes are united, we are getting zygote. So in the zygote and before the gamete formation, the genes are same type of order, same type of distance is maintained. So the tendency of the genes remain together in their original combination during inheritance is called as linkage. This phenomena of linkage was first reported by Bateson and Punnett in 1906. For example, we can see here, this is the one chromosome. So in this chromosome, number of genes are arranged sequentially. A is there, here B is there, here C is there, here D is there, for example. So some genes are very close togetherly. some genes are relatively somewhat distance is there in between the two genes. So the genes are not altered. They as usually enter into the gamete, and the same type of combination will be seen in progeny also. That kind of tendency is called as togetherness of genes are called as లింకేజ్. సహలగ్నత అంటే జన్యువులు లంకెపడి ఉండడం అన్నమాట. జన్యువులు అతి దగ్గరగా అతి సమీపంగా ఉండి లంకెపడి ఉండే ప్రవృత్తినే మనం లింకేజ్ అని చెప్పి పిలుస్తాం అన్నమాట. సో ఫర్ ఎగ్జాంపుల్ హియర్ వన్ జీన్ ఇస్ దేర్, here another one gene is there. There is no linkage we can observe because the two genes are arranged on different chromosomes. And here one gene is here, another gene is very far away from the gene 1. So that's why there is no such type of linkage, more linkage not observed in these two genes. So these, in this case we can call as not linked, in this case also we can call as not linked. So but in this case they are linked genes, confined as linked genes. Is it clear? Okay.
Yes, sir.
Next slide you can go for. So what is the characteristics of linkage? So genes that show linkage are situated in the same chromosomes. Whatever the genes are present on the chromosomes, those genes only exhibit the linkage phenomena. So genes are arranged in a linear fashion in the chromosome. So we can expect the genes are arranged in a linear line. The distance between the linked genes is inversely proportional to the strength of linkage. So we can write one formula here. So what is that? Strength of linkage. Strength of linkage is inversely proportional to distance between the genes. సో రెండు జన్యువుల మధ్య దూరంని బట్టి సహలగ్నత బలం అనేది నిర్ణయించబడుతుంది. ఫర్ ఎగ్జాంపుల్ ఇన్ ఎ క్రోమోజోమ్ ద టూ జీన్స్ ఆర్ వెరీ క్లోజ్ టుగెదర్. ఫర్ ఎగ్జాంపుల్ హియర్ దీస్ టూ జీన్స్ ఆర్ దేర్. ఇన్ దిస్ కేస్ వెరీ క్లోజర్ జీన్స్ షోస్ స్ట్రాంగ్ లింకేజ్, స్ట్రాంగర్ లింకేజ్ ఈస్ దేర్. ఎప్పుడైతే రెండు సమీపంగా ఉండే జన్యువులు ఉంటాయో వాటి మధ్య సహలగ్నత బలం విలువ ఎక్కువ ఉంటుంది అన్నమాట. In another chromosome, relatively somewhat the distance between the genes is very far. In this case, the strength of linkage is weak. అంటే దూరంగా ఉన్న రెండు జన్యువుల మధ్య సహలగ్నత బలం విలువ తగ్గుతుందన్నమాట. అంటే ఇన్వర్స్లీ ప్రపోర్షనల్ అని గుర్తుపెట్టుకోండి. సో ద స్ట్రెంగ్త్ ఆఫ్ లింకేజ్ ఈజ్ ఇన్వర్స్లీ ప్రపోర్షనల్ టు distance between the two genes. So the distance between the two genes are very near, the strength of linkage is more and very strong. They never separate. They always inherited continuously generation to generation. So in the same way, the distance between the two genes are very far. It may be separate. That means we can observe the strength of the linkage is very less comparatively previous. So in this way you can understood in any chromosome containing many genes, the distance between the genes are vary. So where the two genes are very close, the strength of the linkage is more. Where the distance is very far, we can say that is a weak linkage strength is there. We can easily understood. And how these linked genes arranged on the homologous chromosomes. So if the two dominant genes coming from the single parent, for example this is a male chromosome, this is female chromosome, so these two are homologous chromosomes. So for example A and B are linked genes. The two dominant genes are received from single parent. Such type of arrangement is called as cis arrangement of genes. Two recessive genes are coming from a single parent, that is also comes under cis arrangement. So how the two linked genes are arranged on the chromosome? If any gamete receive the two dominant genes from single parent or two recessive genes from the single parent, that arrangement is nothing but cis arrangement. When we can call as trans arrangement, if one male chromosome consisting one dominant gene, another one is recessive gene. Another female chromosome containing one recessive gene, another dominant gene. Here, two dominant genes are received from two different parents. And one dominant gene received from male side, another dominant received from female side. So such type of arrangement we can call as what? Trans arrangement. Is it clear?
Yes, sir.
Yeah. So based on the linked genes, how they are transmitted from parent. A single parent send the two dominant genes, or two parents are send the dominant genes. So based on that, we can easily predict the arrangement of the dominant genes, linked genes, whether it is in cis condition or trans condition. Okay. So what is types of linkage? Generally, linkage is generally classified on the basis of three criteria: crossing over, how many genes involved in, and how chromosomes, how it involved. So based on these three criteria, linkages are classified in different types. So based on that, crossing over, if crossing over takes place during the meiosis, we can observe in the prophase 5 subphases are there. In the pocketing stage, two homologous chromosomes are arranged in a pair, and these two chromosomes divided into 4 chromatids, that is called tetrad. In the tetrad stage, the two non-sister chromatids, arranged in X-shaped structures, you know very well. For example, this is a inside chromatid. So these X-shaped structures are called as chiasma. So for example, in the chiasma region, crossing over takes place. Crossing over is a phenomena. It is exchange of two chromatids in the homologous chromosomes during the meiosis, cell division. But how we can explain based on the crossing over types of linkage? Complete linkage means what? So if the complete linkage means whatever the genes present on the parents, okay, there is no rearrangement, as it is parental combinations we are expecting in the progeny. Same type of parental characters we can expect. No crossing over takes place. Such type of linkage is called as complete linkage. Complete linkage అంటే ఏంటి అంటే ఏ రెండు జన్యువులు అయితే చాలా అతి దగ్గరగా ఉండి వాటి మధ్య క్రాసింగ్ ఓవర్ జరగకుండా నిరోధించబడుతుందో అప్పుడు ఆ రెండు జన్యువులు F2, F3, F5 ఇలా అన్ని జనరేషన్స్కు కంటిన్యూస్గా ఏమైతుంటే సెండ్ చేయబడుతుంటాయి. దట్ ఈస్ నథింగ్ బట్ కంప్లీట్ లింకేజ్. అంటే ఇక్కడ ఎప్పుడు కంప్లీట్ లింకేజ్ అంటాం? వెన్ ద absence of crossing over between the two linked genes. If no crossing over, we only expect parental characters. We only expect what type of character? Only parental. Whatever the parents exhibit the characters, those characters are manifest by F1 progeny or F2 progeny, like that. If any character is differ from the parent, that is indicates a crossing over phenomena takes place between the linked genes. అంటే రెండు జన్యువుల మధ్య సహలగ్నత అనేది 100% ఉండి క్రాసింగ్ ఓవర్ లేకుండా ఉంటే అప్పుడు దాన్ని మనం కంప్లీట్ లింకేజ్ అంటాం. కంప్లీట్ లింకేజ్ ఉన్నప్పుడు we can't expect any non-recombinant types. అంటే we never expect recombinant types of gametes. Recombinant type of gametes అంటే ఏంటి ఇక్కడ? ఫర్ ఎగ్జాంపుల్ ఇలా వచ్చింది అనుకో ఒక గ్యామెట్, దట్ ఈస్ రికాంబినెంట్ టైప్. ఇలా వచ్చింది అనుకోండి ఒక గ్యామెట్, దిస్ ఈస్ కాల్డ్ రికాంబినెంట్. యాక్చువల్గా పేరెంట్స్లో ఎట్లున్నాయి, ఇది కాంబినేషన్, పేరెంటల్ కాంబినేషన్. సో ఇక్కడ క్రాసింగ్ ఓవర్ జరగడం వల్ల క్యాపిటల్ ఏ ప్లేస్లో స్మాల్ ఏ వస్తుంది. ఓకేనా? అదేవిధంగా ఇక్కడ స్మాల్ బి ప్లేస్లో మనకు వైస్ వర్సా ఎక్స్చేంజ్ ఆఫ్ ద జీన్స్ టేక్స్ ప్లేస్. అంటే క్రాసింగ్ ఓవర్ జరగడం వల్ల ఏమొస్తాయి మనకు అంటే రికాంబినెంట్ టైప్ ఆఫ్ గ్యామెట్స్ వస్తాయి. రికాంబినెంట్ టైప్ ఆఫ్ గ్యామెట్స్ వచ్చినప్పుడు అలాంగ్ విత్ పేరెంటల్ క్యారెక్టర్స్ we can see some non-parental characters also. That indicates what? Incomplete linkage or partial linkage. In a chromosome, two linked genes are separated due to the crossing over. If the crossing over occurs in the linked genes, it produce non-parental type, or recombinant type of gametes. These recombinant gamete type of gametes united with the normal gametes, we are getting a different character in the progeny. సో ఎప్పుడైతే తల్లిదండ్రుల లక్షణాలు కాకుండా భిన్నమైనటువంటి నాన్ పేరెంటల్ క్యారెక్టర్స్ కనుక వచ్చినట్లయితే అది ఇండికేట్స్ వాట్ అంటే ఇన్కంప్లీట్ లింకేజ్, అసంపూర్ణ సహలగ్నత. సంపూర్ణ సహలగ్నతలో క్రాసింగ్ ఓవర్ జరగదు, 100% పేరెంటల్ క్యారెక్టర్సే వస్తాయి, వేరే నాన్ పేరెంటల్ క్యారెక్టర్స్ రావడానికి స్కోప్ ఉండదని గుర్తుపెట్టుకోండి. ఇజ్ ఇట్ క్లియర్? ఇజ్ ఇట్ క్లియర్?
Yes, sir. Yes, sir.
యా. సో ఇన్కంప్లీట్ లింకేజ్లో నాన్ పేరెంటల్ క్యారెక్టర్స్ రావడానికి మనకు అవకాశం ఉంటుంది. ఓకే, నెక్స్ట్. సో టైప్స్ ఆఫ్ లింకేజ్ బేస్డ్ ఆన్ ద జీన్స్ ఇన్వాల్వ్డ్. ఇంతకుముందు చెప్పింది ఇది కప్లింగ్ ఫేజ్, రిపల్షన్ ఫేజ్. డామినెంట్ అలీల్స్ ఆఫ్ సమ్ జీన్స్ ఆర్ లింక్డ్ విత్ రెసెసివ్ అలీల్స్ ఆఫ్ అదర్ జీన్స్ ఆన్ సేమ్ క్రోమోజోమ్ షోస్ రిపల్షన్ ఫేజ్. అంటే ఒక క్యాపిటల్ లెటర్ జీన్, ఒక స్మాల్ లెటర్ జీన్ కనుక క్రోమోజోమ్ పై ఉన్నట్లయితే దట్ ఇండికేట్స్ రిపల్షన్ ఫేజ్. టూ డామినెంట్ జీన్స్, టూ రెసెసివ్ జీన్స్ కనుక ఉన్నట్లయితే దట్ ఈస్ కాల్డ్ కప్లింగ్ ఫేజ్. The linkage phenomena explained by these two terms, coupling phase and repulsion phase. Repulsion phase is nothing but the genes, two linked genes are in transposition. In coupling phase, the two linked genes are in cis position. Cis ante two dominant genes oka parent nunchi vastayi. Repulsion phase ante two dominant genes different parent, two parents nunchi vastayi. So that is indicates based on the genes involved type of linkage. Okay, next. Types of linkage based on the chromosome. What kind of chromosomes involved in the linkage? Whether it is autosomes or allosomes. If the allosomes are involved, that is nothing but X and Y chromosomes. Autosomes nothing but somatic chromosomes. శారీరక క్రోమోజోములు ఇన్వాల్వ్ అయినాయా లింకేజ్ లో, లేకపోతే సెక్స్ క్రోమోజోమ్స్ లో లింకేజ్ ఇన్వాల్వ్ అయిందా అన్న దాన్ని బట్టి మనకు ఈ రెండు రకాల లింకేజ్ చెప్పబడతాయి. ఆటోజోమల్ లింకేజ్, అల్లోజోమల్ లింకేజ్. దీంట్లో పెద్దగా మనం చెప్పుకునేది ఏమి ఉండదు. సో ఇన్ ఎవ్రీ ఆర్గానిజం హౌ మెనీ లింకేజ్ గ్రూప్స్ ఆర్ దేర్? సో హియర్ లింకేజ్ గ్రూప్స్ present in an organism which is equivalent to haploid set of chromosome number. Are you getting my point? How many linkage groups present in a organism? That number is equivalent to haploid set of chromosomes. ఒక జీవిలో ఎన్ని సహలగ్నత క్రోమోజోమ్ సమూహాలు ఉంటాయి అని అడిగితే ఆ జీవి యొక్క ఏకస్థితిక క్రోమోజోముల సంఖ్యకు సమానమని చెప్పాలి. దట్ మీన్స్ if the haploid set number of chromosome is equivalent to linkage groups present in the organism. Are you followed?
Yes, sir. Yes, sir.
In the case of humans, how many linkage groups are there?
23, sir.
23. In Pisum sativum, 7. Okay.
7.
In maize, 10. Because 2x is equal to 20, x is equal to 10. So in Drosophila, we can say that is 4. We can hear linkage groups. So in this way, you can any competitive examination ask how many linkage groups that indicates haploid set of chromosomes. Okay. Next. What is the significance of linkage? So this linkage is ensured to keep the genes in a chromosome to inherit togetherly. అంటే ఎక్కువ ప్రాజనీలో పేరెంటల్ క్యారెక్టర్సే కనబడుతున్నాయి అంటే డెఫినెట్గా అక్కడ లింకేజ్ గ్రూప్స్ ఉన్నట్టు, లింకేజ్ జీన్స్ ఉన్నట్టు, లింక్డ్ జీన్స్ ఆర్ దేర్ ఇన్ ద క్రోమోసోమ్స్. Linkage play an important role in determining the nature of scope of hybridization and selection programs. Linkage reduces the chance of recombination of genes and thus help to hold parental characteristics together. It thus helps organism to maintain its parental, racial and other characters. And this linkage phenomenon is also helps in to construct the genomic maps or physical maps or chromosomal maps, which illustrate the distance between the genes and also position of the genes and also relative distance between the genes and gene order also. We can explain in the next classes. So next, what is crossing over? Crossing over, you know very well, it is studied in the meiosis, cell division. It is the exchange of two small segments between non-sister chromatids of homologous chromosomes. The term crossing over was first time coined by Morgan. It is the mutual exchange of segments of genetic material between non-sister chromatids of two homologous chromosomes so as to produce recombinations or new combination of genes. So these are the two homologous chromosomes are there. When the two non-sister chromatids overlap and form a X-shaped structures, that is nothing but chiasma. In the chiasma, the fragments of the non-sister chromatids break down and reunion takes exchange with each other and reunion takes place. That is nothing but crossing over. You know very well, two type of enzymes are involved in this process. Endonuclease enzymes are helps in the breakage of chromatid fragments and the ligase enzymes helps in the reunion of the chromatids. So that's why we are getting violet color chromosome receive a small purple color chromosome, and a purple color chromosome also receive a small portion of violet chromatid. Yes. And this crossing over takes place in which stage? Pocket-in stage. Which stage? Pocket-in stage in the meiosis 1. So chiasmata represent the site of crossing over, where it is exact location of the crossing over during the cell division. That is chiasmata. It is the point at which two homologous non-sister chromatids exchange genetic material. So kinds of crossing over. This is very, very important. You can understood the 2-point test cross problem, 3-point test cross problem based on this only. So kinds of crossing over, depending upon the number of chiasmata appeared, kinds of crossing over we can be classified like this: single crossover, double crossover, multiple crossover. What is meant by single crossover? Single crossover is nothing but only one chiasma is formed, which leads to formation of single crossover gametes. It is most common type of crossing over. In most of the organisms, in most of the plants, single crossover takes place commonly. So just I will go through the diagram. This is a chromosome. This is another related haploid chromosome. Here A gene is here, B gene is here, C gene is here. So respect to homologous chromosomes containing recessive genes. Okay. So the crossing over takes place with reference to one point. With reference to one point, they can exchange the genes from this place to this place. This is called SCO, single crossover. Is it clear? With reference to one point, the chromatids are exchanged. After exchange, what you expect here? Here capital A, here small b, here small c. Here small a, here capital B and capital C here. This is single crossover. When we can call as double crossover, two chiasmatas are developed. This chiasmata may appear between the same chromatids or between the different chromatids. This type of crossing over forms double crossovers. For example, with reference to 2 points, So, for example, here B is there, no? Here small b is there. If they are exchanged, both the genes are exchanged with reference to 2 points. With reference to 2 points, the portion of chromatid exchange that is called as double crossover. That is called double crossover. When we can call as multiple crossovers, many chiasmatas are formed. The genes are exchanged with reference to more than 2 points at a time. But it is very, very rarely occur. Commonly occur which type of crossover? Single crossover. Sometimes there is a scope for double crossover also. But multiple crossovers are very, very rare. It may not happen naturally. So here more than 2 chiasmatas are constituted. It may be further classified into triple or quadruple and so on. So multiple crossing over is of rare occurrence. Is it clear?
Yes, sir.
Okay. Oh. Okay, I will clear the drawings. Okay, next slide. So mechanism of crossing over, so many are there. Stern and Hota. There is a breakage of chromatid segments and exchange of the non-sister chromatid segments, and later their fusion in new places. How this crossing over takes place between two non-sister chromatids. So many different mechanisms are there. Cytological basis of crossing over, and Stern stress and strain theory is also there, but need not to go for that theories. We can go for factors influencing crossing over. The crossing over is influenced by distance between the genes, age, and gender or sex of the organism, and some mutants also, temperature and chemicals. So these are the various factors which are increase the rate of crossing over, sometimes prevent the crossing over also. So increases, if it is increase the crossing over rate, what happened? We are getting more recombinant types. So these are the factors influencing the crossing over. So age, increase in age decreases the degree of crossing over. Male Drosophila shows little crossing over. In case of female, it is more comparatively. And exposure to X-rays increase the incidence of crossing over. And a longer exposure to the X-rays, which leads to influence or which will be induce the crossing over percentage among the chromosomes. And also temperature is also increases temperatures, it increase the frequency of crossing over. Chemicals also. Next, what is the significance of crossing over? Crossing over produces a new combination of genes which is alter the parental combination. Crossing over వల్ల ఏం జరుగుతుంది? పేరెంటల్ లో ఉన్నటువంటి జీన్ ఆర్డర్ చేంజ్ అవుతుంది, ఆల్టర్ అవుతుంది. ద ఆల్టర్ సీక్వెన్స్ ఆఫ్ ద పేరెంటల్ జీన్స్ డ్యూ టు ద క్రాసింగ్ ఓవర్. Crossing over play an essential role in the process of evolution. How the new species are arrived. So we can know very well the significance of meiosis. By the resulting of meiosis, recombinants are formed. The number of recombinants are more, the definitely it increase, it will be induce the different type of species are originated in future. So that is helps in the evolution process. And the crossing over frequency helps in the construction of genetic maps. Crossing over provides inexhaustible store of genes, variability in sexually reproducing organisms, and useful recombinations are used by the plant and animal breeders. If any character is beneficial, definitely the human being catch that character and improve the variety and release into the market as a new variety by the plant breeder or animal breeder. And the origin of a new character also. So this crossing over is also necessary for natural selection. If due to the crossing over a new character is obtained, that will be versatilely applicable and also accepted by the nature, definitely it increases the chance of variation. So green revolution has been achieved in India due to this selective picking up useful recombinants. Operation Flood or white revolution is also being carried out the similar lines. So what is the difference between crossing over and linkage? Crossing over, it leads to separation of linkage. Crossing over ఎప్పుడు linked genes ని విడగొట్టాలని చూస్తుంది. లింకేజ్ ఏమో పేరెంటల్ జీన్స్ యాజ్ ఇట్ ఈస్ గా ఉంచాలని ప్రయత్నం చేస్తుంది. ఇట్ కీప్స్ ద జీన్స్ యాజ్ యూజువల్ టుగెదర్లీ. సో ఇట్ ఇన్వాల్వ్స్ ద ఎక్స్చేంజ్ ఆఫ్ సెగ్మెంట్స్ బిట్వీన్ ద నాన్ సిస్టర్ క్రోమాటిడ్స్ ఆఫ్ హోమోలోగస్ క్రోమోజోమ్. ఇట్ ఇన్వాల్వ్స్ ఇండివిజువల్ chromosomes. The frequency of crossing over can never exceed. This is very, very important. In CSIR examination, under 2 marks, this question is asked like, in any organism, the crossing over percent is never beyond 50%. We can't expect more than 50%. It is always below the 50%. The number of linkage groups can never be more than haploid chromosome number. It increases the variability by forming new gene combination. It reduces the variability. It provides the equal frequency of parental and recombinant types in test cross progeny. It produces higher frequency of parental type. ఎప్పుడు కూడా గుర్తుపెట్టుకోండి, ఏదైనా ఒక పేరెంట్ మధ్య లింకేజ్ ఎక్కువ ఉందనుకోండి, అప్పుడు పేరెంటల్ ఫ్రీక్వెన్సీ ఎక్కువ వస్తుంది. హయ్యర్ 90%, 99%, 100% పేరెంటల్ సిమిలర్ ప్లాంట్స్ వస్తాయి టెస్ట్ క్రాస్ చేసినప్పుడు. కానీ ఎక్కడైనా క్రాసింగ్ ఓవర్ జరిగిందంటే అక్కడ మనము పేరెంటల్తో పాటు రికాంబినెంట్ టైప్స్ వస్తాయి. దీన్ని ఎట్లా కనుక్కోవచ్చు మనం? వెన్ వి F1 plant test crossed, if F1 plant test crossed, the progeny shows only parental characters, that indicates 100% linkage. If the progeny shows along with parental characters few recombinants are there, definitely few percentage or a little amount of crossing over takes place in any homologous chromosome. That indicates. Is it clear?
Yes, sir.
Yeah. So this is about introduction for importance and scope of linkage and crossing over. Tomorrow we will explain about 2-point test cross and 3-point test cross problems. Is it clear for everything?
Okay, sir.
Okay. I will call those. Sir is there? Sir, Tirupati sir.